Good day, everyone, and thank you for joining the H.C. Wainwright 23rd Annual Global Investment Conference. My name is Vernon Bernardino, and I'm a senior analyst at H.C. Wainwright. While we are in virtual format this year, we're confident we can provide value to you with over 850 companies presenting and available for one-on-one meetings. We recommend you meet with all of our companies as each has a special story that you should hear. With that said, I'd like to introduce you to Dyadic, a company that has a very special protein production platform called C1. With us today, our CEO, Mark Emelfarb. With that, Mark, please take it away.
Great, thanks, Vernon. And I'm excited here to be at the H.C. Wainwright conference for a lot of reasons. I think importantly, we are addressing what we believe is a big gaping hole in biomanufacturing. And we're addressing the vaccine and drug shortfalls that the pandemic has highlighted to all of us. Safe harbor statement, again, this shows that some of the statements we make today are forward-looking. So our mission at Dyadic is to transform biomanufacturing. And when we talk about transforming biomanufacturing, that's what we're talking about here is the ability to produce biologic drugs, vaccines, and other biological products, and enzymes and other processing aids in the processing of such processes of biological products as well. And it's not just tied into animal and human health, which is what our major focus is. If we recall, we used to be in the industrial biotech business, feeding and fueling the world, in agriculture, which is now exploding as well. And we're addressing and evaluating where and how to play in that space, and if we want to do that amongst all the other opportunities. So Dyadic at a glance, we have a very strong balance sheet, $25.8 million as of the last quarter. We have no debt, no warrants. And again, as Vernon pointed out, we have a very powerful proprietary and patented biologic vaccine and production platform designed to bring drugs and vaccines to market quicker in greater volumes at lower cost. Investment highlights: versatile platform, strong competitive advantage, large addressable markets, which we'll get into, ready for disruption, transformation, strategic partnerships in animal health, human health, and in other industries and applications are growing and building, and a very experienced board and management team and leadership team. The management team is very small, but very focused. So we have our eyes on the ball and we're moving the ball down the field. Myself, founder, CEO of Dyadic, largest shareholder, approximately 30% between me and my family's trust. Ping Rawson, CFO, Ronan Chudith, CSO. You can see the backgrounds are very strong. Matt Jones, Chief Commercial Officer. And our board is really phenomenal. Dr. Bose from Pfizer, Biologics, Dr. Buckland from Merck. Both of them spent nearly 3 decades. He's more of a vaccine guy, Dr. Buckland. Dr. Bose more an antibody and therapeutic protein, but they both have experience on both fronts. Mike Tarnock, who actually worked at Pfizer for over 3 decades, the chairman of the board, was also the chairman of Carex. And Pat Lucy with Phoenix, which was sold last year to Ligand for over $500 million. And the relevance thing there is the Ligand purchase was about a microbial bacterial fermentation process, which was Pseudomonas, versus what we're using, which is a filamentous fungi glycosylated cell line that has much broader, deeper, more, we believe, valuable applications. So that was a $500 million acquisition. We think GlycoFi was $420 million. Crucell was a couple billion a few years back with J&J. So we can go on and on, but the opportunities to address the markets are broad and diverse. The history of C1, I'm not going to go through the dates and the timeline. I think it's important that you guys realize this is nearly 3 decades of development of this technology. And the technology was developed with serendipitous mutations, random mutations, site-specific integration, synthetic biology, just about everything you can imagine to bring something to bear upon the problems of biomanufacturing in a more efficient, faster way that can be produced in standard E. coli fermenters. And we're heading towards our first human trial of a vaccine or a protein produced from the C1 platform. On the SARS-CoV-2 front, or coronavirus front, we are developing a vaccine called DYA100. We're doing it in conjunction with people like Sorrento Therapeutics, which we announced recently, which is providing us with a $5 million cash upfront payment, $5 million in stock, and up to $4 million in reimbursements. On top of that, $33 million in potential milestones and royalties. And that could be quite large, quite big, and covers a big portion of the globe. In addition, we're working with Syngene that was announced. And Syngene is a CDMO like Lonza, WuXi, Catalent, and Patheon. They're one of the best and the brightest. They work with Big Pharma for over, I think, almost 2 decades now. And they can help us develop and move along COVID-19, trivalent, tetravalent, or monocomponent valent vaccines. Not only both with Sorrento and Syngene are also interested in the platform and how it can be used to develop other protein therapeutics as well as vaccines other than COVID-19. Syngene is also owned 70% by Biocon. And most of the Americans don't know Syngene and Biocon. And I would say you should dig in and do your exploration because they're both very large, experienced companies in therapeutic protein development and production and commercialization. Things like insulin, for example, and monoclonal antibodies. Meditox, 4th largest company in the Botox space. As Dr. Yang pointed out last year in the press release earlier this year, they've been working with us since July 2020. They tried CHO cells, they tried insect cells or baculovirus cells. They saw that the C1 technology produces far more quicker, faster, cheaper proteins and antigens, and they're looking at it not again just for COVID, but potentially for other things. And then the Rubik Consortium in South Africa completes the puzzle where we're nearly 100% coverage on these relationships for coronavirus vaccines. And in the case of Rubik, they're also talking about potentially building a vaccine manufacturing facility in South Africa to serve the African continent. And not just for COVID-19, but to develop other vaccines and proteins using C1 in conjunction with Dyadic. And so if you think about it, in Africa alone, 1% of the vaccines are produced in Africa and 99% are produced outside. So they need our help. And this is going to be a great opportunity to really bring the cost down and the availability to that continent. So again, pointing out this map just highlights the global coverage. And this is on the coronavirus programs, but we're in discussions with people on seasonal flu, malaria, Zika, and other things as well. So I think that you guys should stay tuned because there's a lot of opportunities that we're going to be bringing forth. The development pipeline, I'm not going to walk through this. You can look at it. We just talked about Syngene, Meditox, Rubik. We worked with ID Biologics, which is working with the antibody for SARS-CoV-2 that came out of Vanderbilt, Jim Crow's lab, and Epigen. If you remember, we talked about last October, about a year ago, that they're looking to get funding to move that program around. And I'd say stay tuned and let's see where that goes. On the human side, non-COVID, Hengrui, Chinese, largest, I think, by market cap, Chinese biologic drug company or biologic company. Turtle Tree, we're working on a variety of different things there. Other top pharma companies. We're working on the influenza vaccine program with the University of Oslo. We'll talk about that later. And other collaborations, both in animal health and human health, including moving into the second stage of a program for respiratory disease for birds. And then, of course, the manufacturing problem exists all throughout the biotech industry, whether it's animal health, human health, vaccines, biosimilars, biopharmaceuticals, enzyme catalysts, antibodies. You know, the sky's the limit. And they really have the same problem. How do you scale large quantities of affordable products, either for sale as a product itself, a bioproduct, or as a catalyst used in the manufacturing of those in the processes? So the markets are huge, they're enormous, they're growing. And as you can see here, we're talking about $300, $400, $500 billion, almost $500 billion in market opportunities. And this isn't even all of them. This is just the ones that are defined. If we just get a small fraction of one of these, let alone a small piece of all of them, you're talking enormous opportunities for this company and for the world at large. So what is C1? C1 is a thermophilic filamentous fungi that's been engineered over 20 plus years. It's commercially proven. It grows in very large quantities at very low cost. We used it in the industrial sector for over 2.5 decades, scaled up to 500,000 liters. And using C1 cells in biomanufacturing and animal and human health is poised to transform healthcare. It's poised to make access and affordability a reality. And we're starting to do that. We're starting to see that. The acceleration, the adoption, and the use of C1 is starting to grow. And we're ready to deploy it. And we're deploying it already on the antigen side for vaccines. And we're developing the glycoengineering of the C1 cell lines for therapeutic proteins like monoclonal antibodies, things like KEYTRUDA, which is a $1.5 billion Merck cancer drug, Humira, Enbrel, EPO, all brand name drugs, potentially new drugs, potentially biobetters, and other opportunities. And we already have the US FDA GRAS status on an enzyme produced in C1 toxicology study. So far as we reported last, looks very, very well. And we expect sometime later this month, beginning of Q4, to get the final data, which would be a check mark to move towards advancing first human clinical trial. And there's no need to commercially retool the fermentation. The purity, the productivity, the robustness, the speed, the cost, these are just some of the competitive advantages of C1. C1 also is a filamentous fungi. It's very unique and novel in the way it grows, its robustness, its versatility, the lack of non-sporulation. Other filamentous fungi sporulate. They can't be used under CGMP. We're already producing the antigen in Israel with BTG under CGMP conditions. Antibodies. It's not all about COVID. It's not all about antigens. We can also potentially make monoclonal antibodies. $200–300 billion market opportunity today and growing. We can do 3 or 4 batches in the same time it takes to make one batch of a CHO cell. The cost to produce it is lower. The production time is faster. Our doubling time is 2.5 hours versus approximately 24 hours for CHO. The yields are potentially higher. So we can revolutionize and transform the biomanufacturing of potentially therapeutic proteins, including bispecifics and mAbs, et cetera. And this is just a taste of many of the different types, quantities, qualities, varieties of proteins to be produced at record levels at C1. And we haven't even optimized many of these processes. These are just sort of the beginning. So we're hitting these very, very high levels. In the enzyme space, we actually made one protein at 80 grams per liter. So there's a lot of room for margin. So if you think about it, we're far exceeding the yields of cell lines that they've been using for 3.5 decades, where there's been billions of dollars spent on them. So we have a lot of running room to make quicker, faster, better in terms of yield, productivity, and quality by using synthetic biology. But it's good enough. And in fact, it's better than most. In the case of antigens, I believe we have the most robust, versatile production system for recombinant protein antigens compared to anybody on the planet. We've developed the technology in the industrial space for 2.5 decades. Over the last several years, 5+ years, we've been engineering it to go into animal and human health. The ZAPI program we've talked about, you can look on our website. It's a Zoonosis Anticipation Preparative Initiative. Their goal was to get ready for a pandemic. Guess what? We were ready. We had the ability to make billions of doses of low-cost vaccines using single standard microbial fermenters of different sizes and varieties and types under CGMP manufacturing. And we're doing that now. In the Israeli Institute of Biological Research, we were a couple of years ahead of the pandemic. It had already proven the use of C1 to make an enzyme to protect them against sarin gas. And we've demonstrated safety, efficacy, and protection in our preclinical animal studies, not only with the Israelis, but a variety of other animal studies all over the earth. Human pharmaceuticals, Opdivo, we're working on a biobetter, biosimilar. We're proving this is our proof of concept. This is an $8 billion drug for Bristol Myers Squibb. If we can make Opdivo, we can make Humira, we make KEYTRUDA. We can make a variety of different monoclonal antibodies, bispecifics, trispecifics. We have a very simple, easy, fast way of engineering these cells. And now we're putting human-like glycan structures on these proteins to mimic the human glycan structure in a similar way that we believe will allow us to transform the biomanufacturing of antibodies and other therapeutics. Business development strategy, big pharma, like we did in the industrial sector when we had BASF, Shell Oil, Abengoa Bioenergy, upfront cash payments, milestones and royalties, small, medium biotechs, potential with a similar upfront cash, milestones and royalties, just like the Sorrento deal. And then of course, on small companies also potentially for equity, like we did with BDI in Spain, where we just now sold our equity for 1.3 million euros. So in that particular case, we got the training, we have a company up and running that can work on C1, they had the experience, they moved into pharma with us, and we got all our money back, 30% return, and $1 million worth of research and development to kick. Internal product development, the DYA100 vaccine candidate, moving towards the clinic, the glycoengineering we just talked about, the nivolumab, proof of concept for production of mAbs in general. We've been working on protease deletions and engineering, and then SARS-CoV-2 variants of concern. We've produced already Wuhan, UK, South African, Brazilian, and now we actually have made first version of the Delta variant as well. And we're working on other antibodies and other metabolites. And so this is just giving you an example, as we mentioned earlier, Ligand purchased Phoenix for $516 million in 2020. That was on a bacterial cell that has much more limited opportunities than a eukaryotic fungal cell. J&J paid $2.4 billion to Crucell. Merck paid $400 million to Glycophi, and Sanofi bought the insect cell baculovirus system from Protein Sciences for $750 million. Now, there were other things that came with those acquisitions, and we believe that our value is equal to the biggest of those in the potential for the long term, and we're working towards that goal and objective. Our strategic objectives here are to advance towards a phase 1 clinical trial for our COVID-2 vaccine on a variety of fronts. One now with Sorrento picking up the DYA100 program, so it cuts our expenses and brings in cash. So it strengthens our balance sheet even further, and it reduces the expenditures going forward. In addition to that, we're working in India, we're working in Africa, we're working in Korea. One or more of those may also move forward to phase 1, phase 2, phase 3 commercialization. If any one of those 4 programs work just in phase 1, it opens the door up to a variety of new opportunities, and the adoption and use of C1 will expand, we believe, exponentially. And the purpose here is just first and foremost to prove and demonstrate safety and preliminary efficacy of a protein produced from this cell line in human beings. And then to expand the opportunities into bringing monoclonal antibodies, therapeutic proteins, and of course, a SARS-CoV-2, a monovalent, tetravalent, trivalent, quadrivalent vaccine to address the variants of concern. On animal health, we're working with several different programs, including acute respiratory disease of birds, which we talked about is now in the 2nd stage of trials. The first 2 proteins look very good, and now they're doing further safety and efficacy studies on those. And on the human pharmaceutical side, we're working with several big pharmaceutical companies. We're working on our own of the glycoengineering, our own potential pipeline candidate, and nivolumab and others. And we've produced bispecifics, trispecifics, et cetera, at very high levels that were active, both neutralization, binding, et cetera. And then the potential here in infectious disease, our platform for animal and human health, think about it. There are more animals on the planet than humans. The needs for potential SARS-CoV-2 vaccines or antibodies for humans and animals could be exponentially a lot bigger than we're looking at today. But the market today for us, if we only got a small fraction of that, would be enormous. We've already have 5 variants of concern. As I mentioned, the RBDs produced from C1, including now the Delta. University of Oslo, that mice trial was safe, effective, and protective in the challenge study, and it supports the previous mice study that Sanofi Pasteur did for influenza for hemagglutinin with C1. And then we're working on a monoclonal antibody, as we mentioned. We've developed one in conjunction with ID Biologics that we believe has the potential to be used for all the variants. And we're working on Rift Valley fever, Zika, and other infectious disease antibodies and antigens. Key takeaways: proprietary C1 gene expression technology, patented, some new patents that actually haven't even been published yet, give us even broader running room, competitive advantages, speed, productivity, cost, ease of use, robustness, versatility, the ability to take this technology and quickly tech transfer it all over the globe in standard E. coli microbial fermenters. An example of that is in the case of Syngene, as I mentioned on our conference call. Within 2 weeks of getting the technology, they'd already hit 80% of the number for productivity that was expected. That's remarkable. So we can tech transfer it quickly to satisfy the global desire and demand and needs of biologic vaccines and drugs. Validating partnerships. We did it once before on the industrial side. Shell, BASF, Abengoa raised over $30-some million in upfront access fees, and then $75 million payment for the biotech industrial business from DuPont, which now, by the way, our non-compete is over with, and we're evaluating on if and how and when and what we may want to approach on the industrial biotech side. Opportunistic business, animal and human health, that's our main focus. Many shots on goal, continuing to have great success on the science and on the business development front. Strong financial position we talked about, it's going to get even stronger. $5 million in cash on the definitive agreement once we get it signed. We now have the binding letter of intent. We had 45 days, so we're in the middle of that, working towards getting the definitive agreement signed. With that, it'll bring $5 million in cash, $5 million in stock of Sorrento stock, and up to $4 million in reimbursement on DYA 100. So strengthening even a strong position already, and a very focused, driven management team. And with that, I want to thank you. And I would say look forward to all the opportunities and the milestones that we have in front of us.
Thank you, Mark. Very exciting stuff.
Thank you.
I want to thank Mark and Dyadic International, as well as all of our presenters, for taking part in what has been a very productive and informative series of presentations. Hopefully our next conference will be one that we can hold in person. But in the meantime, we're very grateful for your flexibility and your online attendance this year. Thank you again from me and the H.C. Wainwright team.